Great. And just to make things more interesting, I've changed the topics a little bit around. So, yeah, I thought I'll have a reference in there. Let's see how this works. So a little bit of an intro, because I feel I really should not be here. I trained as a bench scientist, a lab scientist. I worked in a lab. I did the whole pipetting thing. So that does raise the question, why am I here? And I don't mean this necessarily in the existential way, but well, Neil asked, I'm not very good at saying no. And then I took another look at the agenda. I'm like, okay, I probably know the least of anyone in this room about machine learning. But we've been using machine learning approaches to solve problems now and then. So why don't I show you a few of those cases, as use cases, what we've been playing with, and where those skills come potentially in handy. And there's a second component to that is, so yes, I did work at the bench, but eventually I got very, very bored of those things, particular cell culture, coming in on weekends, doing all of this. So in my case, it wasn't from bench to bedside, but from bench to a desk. I've always been working with computers, so I figured while I was doing my master's, there has to be more. Why can't I start working on the digital side of biology? That sounded like a lovely idea, and the first impulse was, okay, let's work with computer scientists or mathematicians out there and collaborate with them. It's an interesting problem. So I started reading literature, and you come up with things like that, and it's literally from the methods section of a well-received paper that said, we used 3 biologists to annotate our corpus. And I went like, okay, hang on a minute. I do not want to end up in the methods section of our papers in the future. That can't be it. So, yeah, I did actually end up in a PhD project that worked on similar things on text mining. And it meant like quite a lot more doing myself and probably not doing as well as many computer scientists out there. But at least I felt I was a little bit in control of those things. Text mining at that stage, really, really simple, natural language parsing. Coming from a statistics side, you basically just look for words that sound like a disease, words that sound like an enzyme. You try to figure out, is there something in between that links them? You can do it by collocation, distance analysis, all kinds of things. And then every now and then you realize that that very naive approach fails in cases where a disease like renal fibrosis is linked to an enzyme like trypsin, but for the sole reason that people use it as an experimental method. You find many, many other enzymes, luciferase, many kinases and other things that are just being used as part of an experimental technique, have absolutely nothing to do with the disease, so you end up introducing false positives. Again, not unknown problem. In natural language parsing, you can actually start assigning terms to concepts. Concepts stand to certain semantic fields. There's maybe 50 or so around that the NIH or NLM uses. And you figure out, okay, there's a disease, there's a protein, and there's a finding that links them. In the other case, there's an experimental procedure. So what you then have is basically a vector of maybe 50 semantic fields. You can basically just count that, and I started playing around with hidden Markov models, in this case with like a support vector machine. Super simple, really just as an end user, linear kernel, played with that. What I didn't quite realize is at some point I had to generate test data, so I was back to that. At least this time I was doing it to myself, so I did annotate by hand 5,000 sentences, whether they are a true finding or not a true finding, and then fed it into the machine learning algorithm. So that's the background, and that was my PhD. For the past 10 years? 10 years. Jesus, okay. So that was first, I worked with Win, who's going to be the next speaker. I worked for Win first in Cape Town, then in Boston, and with this merry band of people. And for that time, we more and more focused on the sequencing side of things. And for the remainder of this talk, I'm mostly going to focus on projects where we are trying to find systematic differences in samples, differences, SNPs, variation on the genome, ideally that have some sort of functional impact. That means finding letters in an alphabet where your reads that you generate from a sequencer differ from your reference, really. And you can call that big data, you can call it precision medicine, whatever buzzword you really want to be using. At the end of the day, you still have a lot of errors in your data. I know you've had a lot of talks, so I'm really not going to go recap all the sequencing stuff, but one of the reasons is we're still not in a position where we can get very long reads. We're getting better at it, but we definitely can't sequence the whole chromosome, so we have to cut it up, fragment it into small snippets, and sequence those. And those reads, by their very nature, have errors in them. It's again very, very dependent on which technology you're using, whether you're using a single cell technology or a nanopore, or whether you're using something like Illumina. But all of those technologies have certain, sometimes random errors, sometimes systematic errors that confound your analysis. For the sake of argument, let's stick to the Illumina sequencing. First of all, you have amplification artifacts, just PCR artifacts along your way. You have, by the way you spot your DNA, well, it's not exactly spotted anymore, but by the way the DNA is distributed on a flow cell, you have little clusters, and sometimes they're quite pure, so you have quite a bit of a mixed signal in individual reads. But it's also still a biological process, and you have DNA on a glass slide that's being exposed to chemistry, and it just degrades over time. So the longer you take with your process, the less and less signal you'll get out of this. And that's why you get those quality plots where you have like the read position on the x-axis, the quality on the y, and that of course is a very terrible plot. They don't look all like that, but the underlying principle is still there. You lose quality over time when you do those experiments, and that has an impact on your analysis. There's crosstalk between your little clusters if they're too close and you're just still measuring light. And there's something called phasing. And that means, again, it's a biological process how you read those things out. At the first step, you do your first measurement, 99% of your substrate gets modified by your protein or enzyme. 1% doesn't. It either lags behind a little bit or it goes faster and skips ahead. So with each step, you add maybe a 1% error rate. And by the end of your read through a little sequence, your whole signal is actually completely out of phase. That sounds like a machine learning problem. And yes, that's actually one of the papers from Janet Kelso, one of Wynn's PhD students that I had to dig out. It's from 2009, so it's been around for a while. So people started coming up with different models to figure out, well, if I have a nucleotide, a string of characters that is generated by those chemical processes and processed or read by those mechanical or optical processes, what are the error profiles? How do errors accumulate? And then you can model that. You can set training sets, you can simulate data, you feed that into different systems. A couple of cyclic hidden Markov models have been used for that, but also SVMs. Nowadays, you don't realize that those things exist anymore because Illumina just incorporates them into our standard software and they get adjusted automatically for it. Unless you do something very, very specific, you study DNA from ancient mummies or Neanderthal, then it makes sense to generate your own error profiles because there's a different level of errors introduced into those because the DNA is 10,000 years old than what the machines normally see. But by and large, this is something where a fair amount of machine learning algorithms just become transparent to the end user, I'd say. But that gets me to the point that it doesn't matter what you do, you need proper test data and training data to find the outliers. And that's something that we in biology in general really, really struggle with. It's pretty much almost always just simulated data, which is okay if you don't have anything else, but it's usually not as good as the real data. In genomics is one exception to that. It's called, well, a couple of exceptions, but one of the bigger ones is called Genome in a Bottle. It's a consortium driven by Mark Serlet and Justin Zog at NIST, the Institute for Standards and Technology at NIH. They basically ship DNA from one patient or one participant in the 1000 Genome Project, the NA12878. And there's a whole consortium of sequencing providers, technology companies, bioinformaticians like myself, who analyzed that data absolutely to death. It's at least 11, I think it's now 14-time whole genomes. You have lots of exomes. You have Sanger sequencing, Iron Torrent, Solid, Illumina, PacBio. Pretty much everything you can throw, the whole kitchen thing that you have available, has been used on those. Mostly to get a very, very non-platform or non-workflow specific view of what variation should you be able to find if you do your things properly in this particular sample. What this allows you to do is you can actually test new mechanisms, new sequencers, new workflows, or a nice method that you can up with, because there actually now is a test set. And you can ask, well, if I use my very smart way of finding a variant or my aligner, how concordant am I against this whole worldview of other tools out there? That works reasonably well. You can quickly test different workflows for the concordance, sensitivity, specificity. But actually, let me skip forward. I like that. Maybe more importantly is that you can learn something new. So you can write your classifier and say, okay, based on this data, here's my new read data. Tell me which ones you think are likely for true positives and which ones would you filter out because they don't match any of the criteria of the other variants. And as attributes, you can use just about everything you want. You can ask, well, what is the context? What does the genome look like around the position that I call the variant? How many individual observations do I have that support this? Is there any positional bias on where this variant is being called in the reads that I have? Is this only being called in reads where it shows up in the beginning? There's many, many different parameters, probably like 20 or 30 that you can use. Where it gets interesting is when you then take the classifier and you start asking, well, what was the most important metric that made you decide this one is likely a false positive? And one of the things that, for example, comes up is that you normally wouldn't expect for heterozygous calls, in many cases where you have too many independent observations of that event, it's a false positive. It intuitively doesn't make sense. You're like, wait, hang on a minute, I've got 200 observations, but if you only expect 30 observations across your genome on average, and you're like 2 or 3 standard deviations out from there, then something is wrong with the genome build in that position. It's probably something where our genome build isn't very good, where we have multiple genes that are in different places. It just tells you something is wrong. And something you can nicely see, if you have variants, those are true positive, those are false positive variant calls. Those are genomes that are sequenced an average of 80 or 90x. The vast majority have a coverage, like the number of independent observations that are positioned between 100 and 200. There's virtually nothing in the 250 upward space. That's where all the false positives start adding up. Once you have that, you actually don't need the machine learning classifier anymore. You just end up with a hard filter and say, look, anything that has more like 3 standard deviations of the mean coverage, flag them as false positive. Which brings me to the more general problem. In most realistic scenarios, you get a patient, you have some data, you don't have that test data. You just don't. All you get is, here are your 3 million positions where we think your patient differs from our reference. So a standard approach for doing that, again, with machine learning approaches is you just run a whole gamut of different workflows, different aligners, different variant callers, and say, okay, my likely true positive set is the set where every single method agrees. That's underestimates it. You're going to lose a lot of true positives, but at least it's a good enough training set. The negative set is a little more tricky. You can ask, well, anything that's only supported by one specific method, nothing else. But some of them are actually very good in regions where other methods are not as good. So you probably want to combine that. It's like, okay, not just one method and also otherwise weak support. Or you can ask, well, it's a novel variant that nobody in the world has seen before. In other words, it's not in dbSNP, and it also has relatively poor support, probably a false positive. But in both cases, both your true positive and false positive set are going to be noisy. And that maybe explains if you then actually train an SVM on that, and you actually have a couple of million of those events, you only see like an increase in concordance of around 1%. It's still substantial. It's still nice to have. That's still 30,000 variants that you don't have to analyze and comb through later on. But given the time it takes to actually train them and then classify them, all the bias you introduce, we don't really have a good solution for that particular problem just now when it comes to whole genome data and using machine learning approaches to analyze this. Let me move forward. There are other scenarios where it absolutely becomes vital to work with machine learning classifiers. And that's in particular in regions where our tools are just not as good as they are already in the human genome sequencing space. So that's a project that was driven by Jonathan Lee and Dan Koretzka's group on minority variants in HIV. Jonathan studies HIV and particularly is interested in the RNA genome of HIV and how it changes and how resistance to drug treatment actually arises over time. We now have very effective HIV therapy for just about any case. One of those standard drugs is raltegravir. It targets the integrase, the enzyme that allows the HIV RNA genome to integrate into the host genome. It's part of your standard cocktail of 3 drugs that you give to HIV patients. But there's a problem with that. The HIV genome changes way, way, way more rapidly than a human genome. It's very, very error-prone during replication. So you end up quite often when it replicates with cases where random nucleotides just change. And in some cases, those changes are not silent. They're non-synonymous, and you change one amino acid for another. And in some cases, if you're really unlucky, that amino acid change then prevents your drug, raltegravir, from going where it's supposed to be going. So this is the drug in this case, inserted into the integrase enzyme. If there's a change, all of a sudden your effectiveness of that drug to bind your target protein drops dramatically. That means you become resistant to this drug. And the mutation that I showed you is one of those known mutations. If you have that mutation, raltegravir is not the right choice for you. The problem with that is, as I said before, HIV is really, really, really able to change rapidly. It's going to be very hard to see on this screen. The smaller screens are actually going to be slightly easier. That's one of the global influenza pandemics we had. There's like a little tree that shows you all the variants that we found, all the different genome variation, different strains basically that have developed during that pandemic. That's just HIV in Congo in comparison. You see some more diversity in a patient has been chronically affected with HIV for a number of years than you see in a standard flu epidemic, and that's quite massive. In particular, you see minority variants. People get affected normally with one clone. There's one RNA strand that gets infected, but over time, develop mutations at different frequencies. So you might have 60% that have one genotype, there's another 10% has another genotype, 5% another. And that becomes really, really tricky. To the point that we now get standard reports when you do your blood test for an HIV patient, what drug cocktail should you set them on? They screen for all possible and known mutations, all drug resistance, and then come up with a combination cocktail of the 3 drugs that are least likely to clash with existing resistance mutations that patient might already have. So you basically have a number of substitutions that they found already in that patient. That means those drugs, don't use them, are not going to have any impact on that patient. The problem with that, all of that is right now based on Sanger or 454 sequencing. And your sensitivity kind of drops out at around 20%. So any minority variant that is present in a patient's HIV population below 10, 20%, you're not going to pick up. That can be a bit of a problem. In this particular case, it was a problem because there was a Merck clinical trial that failed spectacularly in the sense that a fair number of patients that they gave the drug basically became resistant against it very, very quickly, despite the fact that they screened them before and couldn't find any predisposing resisting mutations. So John asked us, well, can we do this properly? Can we go below 20% using Illumina sequencing? What can we do to figure out, do they have minority variants maybe at 5%, maybe at 1%? Again, having a proper test set is important. John came up with a very simple system. We took 5 different HIV strains, quite a bit different really. Any good old-fashioned way, he mixed them in a test tube at different ratios. So we had like 60% of one, 5% another, 1%, 0.5%. And we knew exactly, because we sequenced those strains, what frequency to expect once we do our sequencing. You mix those, you put the whole thing on a sequencer, you align not against the reference, because that's too diverse. You align against what you would get out of Sanger sequencing on that patient, like a consensus sequence. That helps you quite a bit, really. We did all kinds of other tricks. We tried to smooth the reads because we're sequencing at around 100,000 to 200,000 fold coverage. It's like about 200,000 observations at each site. We spent a lot of time working out realignment because the genome is so diverse. It really makes sense to spend some time in cleaning up the whole alignment. And then we did what I described before. We applied a filtering technique. We looked at our reads and said, okay, when we call a variant at a site, what are the metrics for all the observations at that particular site that support this? What are the base qualities? What are the variant qualities? How well did the read map to that particular position? What's the sequence context, the k-mer context around it? And came up with a very, very simple linear classifier that either kept the read or tossed it, depending on whether it was a yay or a nay. If you do that, and you filter out all the potential sequencing noise, we ended up doing quite well, I thought, because we could actually monitor variance at around 0.5% at that point. The only problem we had is the sites that were supposed to have absolutely no variation. Remember, we know exactly what to expect at each because we mixed our clones. Also varied between 0% and about 0.3%, and it's getting in some cases very, very close to our 0.5 threshold. So it became in many cases difficult to distinguish between the background noise and our true signal at the 0.5% range. Still, we went from 20% to 0.5 or maybe 1%, so I was happy. John wasn't so happy. I was like, well, I think we can do better. We're doing 200,000 observations. There got to be a better way to do this. So we went back to the drawing board, and first of all, we added another clone at 0.1% to be closer to the noise. So we actually also have a true positive set in that range where our noise really was. And then we were like, look, machine learning, we're really not that good at it. So we got kind of lucky. Harvard Catalyst, like a grant project, had funding at the time to support basically outsourcing, or rather crowdsourcing, different algorithms. So if you had a problem that needed an algorithmic solution, they basically donated the prize money, in this case $10,000. You just had to come up with the description. So we spent a fair amount of time abstracting our problem away from the biology. Because again, you don't want to, you have a lot of people who have computer science, math, statistics backgrounds. They don't care about, well, they do care about it necessarily, but you have to make it tangible and tractable for them. So we basically explained in a very simple problem. Look, we are trying to put together, reassemble a book, a text. We have fragments that have a certain error rate. For each position and for each fragment, we have all those different metrics that we can make you available. Your task is to distinguish true variation from any kind of noise that might be in there. We submitted a test set for them to play with, a training set for them to play with. And then the site, top coder, gets a test set. So each time someone submits a solution, they quickly run that and tell the person, okay, your score is now X, Y, Z. People are encouraged to iterate on their solutions. So there's constantly a leaderboard. So over the time we did this, we had around 200 people submitting code, a total of 600 different solutions. So many submitted multiple solutions and iterated on something getting better. But I should say that was version 2.0. Because in the first time, we tried to come up with a nice definition of our problem. They looked at it and went like, hey, that's great. I actually completely can reverse engineer the original strains. So they completely ignored the whole classification problem, just took our test data, re-engineered the original 60%, 5%, 1% strand, and went like, great, anything doesn't fit in there is noise, done. That's not exactly the problem that we were trying to solve. So we had to restart the competition, set another $10,000 of prize money, get a better definition out there, add some noise to our problem. But we got out basically pretty much all of the winning competitors use some variation of a random forest approach. Again, not surprising simply because they're how flexible they are. What did surprise me is we basically supplied them just with 3 metrics, the ones that I mentioned. They combined them, the leading one in this case, into 14 different metrics by combination of them, like linear or non-linear combination of those metrics to have a larger parametric space. Solutions tended to stabilize around 80 to 150 trees, something at that point it didn't really make any difference anymore how much you train. And we got, as part of the competition, the source code, the reference implementation, and everything else. So we tried that and used that to, hold on, no, not 3 microphones, that's one too many. And we used that to go back to original data set, sequence a couple more patients, and looked how well we're doing. We now can pretty much reliably call to the 0.1% metric, roughly. So from 0.5% to 1.1%, with about a 1% false positive rate, which is quite acceptable, really. I think more important is that the classifier they came up with was all of a sudden stable. Before, each time you did a patient and sequenced them, we also had to do our little test experiment on the side because of all the variation that's in between, like a different technicians, different sequencing depth. It was just unstable. So each time you had to retrain it on the test set and then apply it. This, at least in our hands, and the couple of experiments we tried, was stable between experiments. It was not overtrained. It was general enough to be able to handle different cases. So that made us really happy. There's something else that then went on to the clinic, and that's still ongoing, although it took like 3 years by now. It's just a painful process. And I think I constantly underestimate how long it really takes once you think, wait, that's great, that should help patients. 3 years on, we're still in the early stages, because everything you do, the little report that I showed you earlier on that decides whether a patient can get a drug or not, all of that, of course, is FDA approved. The minute you make any change to that, you have like a 3 to 5 year legal process ahead of you, getting the new system showing that it's at least as good, that doesn't do any harm. It's a long process. That said, it's really rewarding to see something like this move into the clinic and then become like a standard process in places like Brigham and Women's, which is where Lynn Bree is. So we'll see whether that actually turns out to be useful. We have some follow-up experiments. One of the little things that we completely ignored here is phasing, meaning we only have little fragments. I can tell you whether there's a variation here, but I can't tell you whether a second variation, a little bit further downstream, is present in the same clone, in the same virus. In many cases, you need like 2 or 3 hits before a strain becomes resistant. And that means you need to have this information whether 2 mutations are actually tightly coupled or not. So that's ongoing work, again, with Jonathan Lee. And all that work was done by Brad Chapman in Wynn's group and my group. Switching topics again, since we were talking about the clinic, one of the nice parts of working in a bioinformatics core is you get to play with so many different data sets. It's just pure joy, really. So this was one of the cases where a pediatrician from Boston Children's Hospital stopped by and said, so this is thing in the ICU where we monitor all our kids constantly. In particular, if you're in a neonatal ICU, kids just stop breathing. They just do regularly. It's apnea, it just happens. It's not a big deal. It happens like during a regular shift, like 2 or 3 kids just stop breathing. It just happens. The alarms go off, the nurse stops by, basically stimulates the kid, either just by tickling their feet, I'm not kidding, or basically taking more severe measures if needed. Usually it's just like 15, 20 seconds where they just forget to breathe. Sometimes it's a little bit more severe. But for them, it's not a case where they panic, it's normal procedure. That said, those kids are hooked up to those monitors pretty much 24/7. So they measure oxygen, carbon dioxide, they measure blood pressure, they measure the heart rate, and more importantly, they don't just measure this as values, but they also measure how it changes, because they measure those things at around between 30 to 60 hertz, depending on what metrics they have. And the newer machines nowadays don't just alert them, but they have little USB sticks, and they store the last 10 minutes of data on there. So you're like, well, wouldn't it be nice if you got an alarm like before the kid stops breathing, rather than after the fact? So we can just walk over, just give them a little shove, and we don't have to worry about it anymore. I'm thinking I have data now collected from over a year from those kids. I have about 500 positive events where I can show you all those metrics, all those waveforms, everything that you need. And I can give you a couple of thousand events where absolutely nothing happened over a certain amount of time. Can you find a better predictor? So that's ongoing work. It's something where we actually pass it on to another group, in this case, dedicated machine learning people. Because at that point I went like, okay, I don't know enough about this. Particular, we need to start thinking about wavelet transforms. For example, are there certain transformations where it's not just an event like, oh, the heart rate goes down or the oxygen drops, but is there a certain pattern or the shape, how the kid breathes in and out, that changes, where the frequency is the same, but it becomes shallower? Are there any other things that we can figure out? So now people over at the Faculty of Arts and Sciences trying to convert that and playing with the data. Which is, I think, the key point. Like, whatever you do in machine learning, there's like an insurmountable number of problems out there. And it's actually, I think that's one of the fun bits. Whatever you pick and working in that domain, there will always be problems for you to tackle. It's just really a matter of going out and talking to the community, talking to people in the hospitals, talking to your colleagues, maybe in departments that you normally would never talk to. I wouldn't have talked to a pediatrician who's an anesthesiologist in children's, because it's not genomics. It's normally not something I do. But he had data, he had an interesting idea, and it sounded tractable and something that you can transform into something that you can then apply standard algorithms to. So I think that's really that part. The only key issue is at the minute you find something that has a good training data set, or you think it's a good training data set that you can generalize on, it's a problem waiting for you to be tackled. That's it from my side.